So as we've already discussed, urine formation occurs in three parts. We previously discussed glomerular filtration, and now we're moving on to part two, which is where we're going to see tubular reabsorption and secretion. In this phase, we convert the glomerular filtrate to urine, and we're going to trace the tubular fluid from the proximal convoluted tubule on through the distal convoluted tubule. So first of all, the proximal convoluted tubule is going to reabsorb about 65% of the glomerular filtrate. And it's going to remove some of the substances to the blood and secrete them into the tubular fluid for disposal in the urine. Tubular reabsorption is the process of reclaiming that water and the solutes from the tubular fluid and returning them into the blood. There are two methods by which we can have tubular reabsorption. First is the transcellular route, where substances pass through the epithelial cells of the proximal convoluted tubule and then are reabsorbed into the blood. And second of all is the paracellular route, where substances are transported between the cells. Solvent drag is what happens when water moves between the cells and it passes through the tight junctions that exist between those epithelial cells, and it drags along with it dissolved solutes. So now we'll examine specific solute reabsorption. So sodium is the primary key to every single other transport because it creates such an osmotic and electrical gradient that it drives the reabsorption of water and other solutes. It's the most abundant cation, that is positive ion, in the filtrate, and it creates a really steep concentration gradient that favors diffusion into other epithelial cells. So there are two types of transport proteins that we're going to examine. And the apical surfaces of the cell. So the apical surface is here on the tubular fluid side. First of all, we're going to see symports such as the sodium and glucose transport protein here. Sodium and glucose transport protein is a symport because it's transporting two things in the same direction, so symport. And then there are other things called antiports, if you remember from general biology. For example, here we have a sodium and hydrogen antiporter. The sodium and hydrogen antiporter transporting sodium into the cell while transporting hydrogen out of the cell. So sodium being the key, let's look at what's going on there first. We have these sodium-potassium pumps, which, as you'll remember, are active transport pumps. They depend entirely upon ATP. So it's highly energy-consuming process. It's going to pump sodium out into the extracellular fluid, which is going to be picked up by the peritubular capillaries, and it's going to be taken away. Because sodium is being transported out by this active transport pump, we're creating a gradient for sodium to come into the cell. So this is secondary active transport, where we'll see sodium and glucose co-transported into the cell. And then we're going to see that negative chloride ions follow this positive sodium ions. And so we have various antiports in the apical cell membrane that can absorb chloride in exchange for other anions, other negative substances that they're going to eject back into the tubular fluid. We also see here that this process acts to remove in the tubular fluid hydrogen ions, which then are going to have an influence on the acidity of the body's tissues. So we're removing hydrogen ions, removing acidity from tissues. So now that we've dealt with reabsorption of sodium and chloride, we have several other electrolytes to pay attention to. Potassium, magnesium, and phosphate ions are going to be reabsorbed through the paracellular method of diffusion, so right between the tight junctions, traveling with water. We'll also see that phosphate is co-transported into the epithelial cells with sodium, and Some calcium is going to be reabsorbed, again through the paracellular route. It's traveling with water. And we'll come back later and deal with more calcium as it's being reabsorbed later in the nephron. Now, this is where all the glucose is normally co-transported with sodium. So all the glucose is going to be reuptaken at this point in the normal situation. However, in situations like diabetes, we're going to see that there's a problem. These are done with glucose is transported with sodium and glucose transport proteins. So it's a co-transporter. We'll see here sodium and glucose transported through the tubular side into the cell. And finally, we have urea. Urea is going to diffuse with water paracellularly between the cells. And here we'll see about 40 or 60% of the urea is going to be reabsorbed. We're going to see the kidneys only remove about half the urea from the blood. So here we will absorb a lot of urea, and then some will actually be reuptaken into the tubular fluid. When we look at creatinine, creatinine is not going to be reabsorbed at all, so it maintains itself in the tubular fluid. And the purpose of the reabsorption is to create some osmotic drive, and we'll see how that comes into play later on. So now, following the reabsorption of all those electrolytes, water will be reabsorbed. As we learned earlier, the kidneys reduce about 180 liters of glomerular filtrate to 1 or 2 liters of urine. So many times the blood volume is actually filtered by the kidneys. And right now, most of it's going to be reabsorbed. 2/3 of the water is going to be reabsorbed here in the proximal convoluted tubule. So reabsorption of all of these other solutes now makes it possible. There's osmotic draw for water from the tubule back into the tissues and into the peritubular capillaries. Water has followed the solutes by osmosis through both the paracellular and transcellular routes. When the water travels through transcellular routes, it's going to move through little things called aquaporins. Aquaporins are embedded in the phospholipid bilayer of the cell. They're little channels that are specific just to water. Here is where we have obligatory water resorption, meaning that the water must be reabsorbed. This amount of water must be reabsorbed. This percentage of water must be reabsorbed in this region. The rest of it later on is reabsorbed. That's going to depend on the level of hydration, so on and so forth. So now we have all these solutes and a lot of water in the tissues surrounding the tubule. So this has created the perfect situation for the peritubular capillaries to uptake all of these goodies. 3 factors are going to promote osmosis into the capillaries. First of all, we have very high interstitial fluid pressure because of the accumulation of reabsorbed fluid and solutes around the basolateral sides, so the tissue sides of the epithelial cells of the tubule. Second of all, we have low blood hydrostatic pressure because we've got narrowing of the efferent arterioles in the peritubular capillaries. And that gives less resistance to absorption because the blood pressure is lower. And third of all, we have an elevated colloid osmotic pressure in the blood. So most of the proteins were not included in the glomerular filtrate, which leaves them in the blood, and thus there's osmotic draw for the fluids out of the surrounding tissues and into the peritubular capillaries. There's a lower blood pressure, BHP, blood hydrostatic pressure, and a high pressure of fluid literally pushing water into the capillaries. So as you probably noticed, there's lots of protein transporters involved in the reabsorption of these various solutes. So naturally, there's going to be a maximum rate of transport of any molecule across the epithelial cells of the tubule, because there's a limited number of transport proteins. And when all those transport proteins are busy, and they're all transporting as much as they can, then anything that's in excess of the transport protein's capacity is going to pass through the kidney tubule. Glucose is of most interest here. So when all the glucose transporters are occupied, the remaining solutes that can't be transported out of the tubule for reabsorption, such as glucose, are going to appear in the urine. With glucose, the maximum that can really be absorbed because of the limited number of transport proteins is about 220 milligrams per deciliter. Anything over that, we're going to see that glucose remains in the tubule and thus ends up in the urine. Often in diabetes, we'll see that glucose levels get up to 400 or 500 milligrams per deciliter. And so we'll see glycosuria, sugar, in the urine. And that's one of the primary keys, the primary signs that diabetes may be an issue. So we've covered reabsorption or reclaiming of stuff, as in solutes and water, from the tubule. Now we're going to deal with secretion of things from the capillaries into the tubule. So things that need to be reclaimed for the urine. So tubular secretion is the process of drawing stuff back out of the tissues and into the tubule, or back out of the capillary blood and into the tubule. So first of all, we need to reclaim a lot of the urea that was reabsorbed in order to create a gradient to move water out back into the tissues and into the capillary blood supply. So now we're going to reclaim or secrete some of that urea into the kidney tubules. Also bile acids, ammonia, catecholamines and prostaglandins. We're also going to see that pollutants are cleared from the blood, things like morphine and penicillin and aspirin. And this is why we need to take those sorts of drugs 2 or 3 times, maybe even 4 times a day, is to keep pace with the rate of clearance of the kidney. Also, tubular secretion is involved in maintaining the acid-base balance. And we're going to revisit this again in chapter 24, the next section of our exploration. So now we move from the proximal convoluted tubule into the nephron loop. And the primary function of the nephron loop is going to be to generate a gradient, a sodium chloride gradient, a solute gradient that enables the collecting duct to concentrate urine and conserve water. Collecting duct is going to happen a little ways down the line. So we're interested in electrolyte reabsorption from the filtrate. And as you already know, there's a thick segment and a thin segment. The thick segment is of importance here. The thick segment is going to be involved in actively transporting sodium, potassium, and chloride out of the tubule. So these ions, sodium, potassium, and chloride, will be transported together into the cell. And then we're going to see that there's active transport with the sodium-potassium pump. So potassium re-enters the cells of the tubule, and sodium is left outside. The catch here is that water can't follow this gradient creation because the thick segment is not permeable to water. So that leaves the tubular fluid very dilute with lots of solutes in the tissue outside of the nephron loop. So now we've completed our journey through the proximal convoluted tubule and the nephron loop. Now the fluid is going to enter the distal convoluted tubule and then finally the collecting duct. So fluid arriving at the distal convoluted tubule still contains quite a lot of water, but only about 20% of the original water that was in the glomerular filtrate, and it has about 7% of the salts. And so we still have lots and lots of water to remove. If this were all passed as urine, we'd still be peeing about 36 liters per day. And you know that we've got to get rid of some more water. So the distal convoluted tubule is where this reabsorption happens. We're also going to see that this is regulated by a lot of hormones. Of note, aldosterone, atrial natriuretic peptide, antidiuretic hormone, and parathyroid hormone. And these hormones are going to act on the principal cells of the distal convoluted tubule. They have receptors for all the hormones, and they're primarily involved in salt and water balance. The other type of cells we'll find in the distal convoluted tubule are these intercalated cells. And they're involved primarily in acid-base balance because they secrete hydrogen ions into the tubule lumen, thus removing them from tissues, and they reabsorb potassium from the solution in the tubules. So now let's examine the effects of these various hormones on the distal convoluted tubule and the collecting duct. We'll start out with aldosterone. Aldosterone is the salt-retaining hormone, and it's secreted by the adrenal cortex. It's the adrenal gland sitting right on top of the kidneys. When blood sodium concentration falls or potassium concentration rises, aldosterone will be signaled to be released from the adrenal cortex. Another thing that might influence aldosterone secretion is a drop in blood pressure, but this influence is indirect. The drop in blood pressure causes renin release from the kidneys, which causes the formation of angiotensin II, and angiotensin II stimulates the adrenal cortex to secrete aldosterone. The functions of aldosterone, primarily it's going to act on the distal convoluted tubule, the thick segment of the nephron loop, and the cortical portion of the collecting duct. So the collecting duct that's in the cortex of the kidney. And it will stimulate the reabsorption of more sodium into the tissues, which will be followed by chloride and water. And it's going to stimulate the secretion of calcium back into the tubules. The net effect of that is that the body's going to retain sodium chloride and water. There's going to be more potassium in the urine. Because water has left the tubule, we're going to see that urine volume is reduced. And we also see that because of these effects, aldosterone helps maintain blood volume and pressure. So aldosterone is the salt-retaining hormone, thus the water-retaining hormone. Now we'll move in to look at atrial natriuretic peptide, which has somewhat opposite effects. Atrial natriuretic peptide is going to have four main actions. It's secreted from the atrial myocardium of the heart in response to high blood pressure. So as blood pressure rises, the main goal is we want to lose more fluid. So all four functions aid in the loss of fluid or retaining fluid in the tubule for secretion in the urine. It's going to first dilate the afferent arteriole and constrict the efferent arteriole, which is going to increase the glomerular filtration rate, thus aiding in the loss of more water. It also acts to inhibit the renin and aldosterone secretion. It inhibits the secretion of antidiuretic hormone, and it inhibits the reabsorption of sodium chloride by the collecting duct. Next, we look at antidiuretic hormone, which is secreted by the posterior lobe of the pituitary. We'll visit this hormone more later, but for now, in response to dehydration and rising blood osmolarity, we're going to see that the hypothalamus is stimulated, which then stimulates the pituitary. The pituitary releases ADH, and the primary action of ADH is to make the collecting duct more permeable to water. And when the collecting duct is more permeable to water, the water in the tubular fluid re-enters tissue fluid and bloodstream rather than being lost in urine. So antidiuretic hormone acts to retain water, antidiuresis, so not to lose water. So finally, we'll take a look at parathyroid hormone, which we've already seen in its role in calcium maintenance. Here, again, its role is in calcium maintenance. It acts to increase calcium retention, lowering calcium content in the urine, and it acts to increase phosphate release into the urine. By having phosphate released and calcium maintained, we'll see that the elevated levels of calcium ions stay in circulation rather than precipitating into bone tissue as calcium phosphate. Parathyroid hormone also stimulates calcitriol synthesis. And this calcitriol synthesis is occurring in the epithelial cells of the proximal convoluted tubule. So in short, parathyroid hormone again is acting to maintain blood calcium levels. So now let's summarize the events of tubular reabsorption and secretion, part 2 of urine formation. So the proximal convoluted tubule is going to reabsorb about 65% of the glomerular filtrate and return it into circulation through the peritubular capillaries. Much of this reabsorption is going to occur by osmosis and co-transport mechanisms linked to some active transport. So lots of ATP is used. And the proximal convoluted tubule is where we have this mandatory amount of reabsorption. Then we have the Nephron Loop, where we're reabsorbing another 25% of the filtrate and setting up the situation, the osmotic situation, for the collecting duct later on. We'll come and revisit that in the next phase. Then we have the distal convoluted tubule, where we're reabsorbing sodium and chloride and water follows. This is where we'll have hormonal control that's dependent on various blood volume, and it's going to influence secretion of all the hormones that we've just discussed. We'll also see here that drugs are extracted, waste, some of the solutes from the blood, and they're secreted into tubular fluids. So this is where we get rid of lots and lots of waste products. So the distal convoluted tubule has now completed the processes of determining the chemical composition of urine. Now the collecting duct in phase 3 of urine formation we'll see is all about conserving water, reabsorbing the final amounts of water using a gradient that's been set up by the Nephron Loop. So now it's time to go to the Connect site and do the Learn Smart module associated with this lecture.